Good afternoon, ladies and gentlemen. I'm speaking to you from Australia, although I'm also present in the room electronically. It's a real pleasure for me to come to this conference. And I came to the conference in China conference on sheep a couple of years ago, and I really enjoyed myself. Today, I'm going to talk to you about the 3 Ps of reproduction: pheromones, photons, and food. And what I'm going to do is take you on a personal journey, my personal journey in science since I was a PhD student back in 1975. At that time, my starting question was, how does the reproductive system of the sheep respond to changes in the environment? And during my career, there have been 3 revolutions. The first is that the brain makes new cells. The second is that we discovered junk DNA, and now we're asking if that was a really good idea or not. And the third revolution was livestock management, and how I in particular have completely inverted my ideas about this. So let's get back to the original question then. How does the reproductive system of the sheep respond to changes in the environment? I'm going to give you a fairly basic overview here of the reproductive system. Many of you will be familiar with this. But basically, reproduction is controlled primarily by the brain through hormones called gonadotrophins, LH and FSH. These hormones control the testis to produce sperm and control the ovary to produce the egg. In addition, of course, the testis produces male sex steroids, and these sex steroids penetrate the whole body of the male sheep and affect male sexual behavior, aggression, horns or antlers depending on the species, neck muscles, mane, and an odor. The odor, as you'll see, I've highlighted it there, and it becomes very important later on. On the female side, we have a similar variety of endpoints in response to female sex steroids: sexual behavior, maternal behavior, the mammary glands, vaginal mucus controlling fertilization, the activity of the uterus, and again, odor. So this system is controlled and affected by 3 major environmental factors. The first is photons, the second is pheromones, and the third is food, and hence my 3 Ps. It's really important to understand here that there's a final common pathway here. The final common pathway in the brain for all of these influences is the molecule GnRH, or gonadotrophin releasing hormone. So this hormone is synthesized in the hypothalamus or preoptic area, the lower part of the brain here, the most primitive part of the brain. And it absorbs information from all around the brain, all sorts of information about the external environment of the animal, the internal environment of the animal. And all of this information the brain brings to the GnRH cells. And these cells then produce LH and FSH that control the testis and the ovary. So basically, we'll need to understand GnRH if we're going to understand this whole process. Let me look a bit more deeply into this then. So the hypothalamus and pituitary gland, I'll blow them up a little bit bigger here so you can see what they look like. And so what you see here is the pituitary gland, the orange object there, and here we have very small blood vessels, portal blood vessels, that bring blood from the brain out through the pituitary gland, transporting pituitary hormones. As I mentioned before, the critical cells here are the gonadotrophin releasing hormone cells. These cells are neurons, and they produce GnRH, and they transport the molecule down the axons and then release it into the bloodstream to control the pituitary gland. Now, some very clever people can actually take blood samples from these little tiny vessels here. And when they do that and measure GnRH, they see this. GnRH is produced in pulses, not as a continuous stream. The GnRH pulses travel into the pituitary gland, and there they stimulate the pituitary gland to produce pulses of LH. In addition, the GnRH controls the secretion of FSH, but it's a relatively uninteresting pattern. The LH and the FSH then control the ovary and the testis. So then what we want to do is understand these cells, the GnRH cells. And they're really hard to study because they're right in the center of the brain. And so really difficult to see. But you can do this. You can kill the animal and you can look at the cells and you can stain the cells, see where they're located, look at the connections. You can even see these days when these cells have been activated or deactivated by clever technologies. But when you do this, of course, the animal is dead. So it limits the sort of experiments you can do. As I said before, some very clever people can actually measure the GnRH in the bloodstream in the center of the brain there. But for the sheep, there are 3 or 4 people around the world who can do this. And basically now they've all retired. I cannot see anybody doing this again for quite some time. But you don't have to do that, because in fact what you can do is measure LH. And because the LH pulses reflect the GnRH pulses, effectively then what you have is a bioactivity, a bioassay of the GnRH cells in the brain. So you can measure LH in very simple blood samples taken from the jugular vein, and therefore understand what is going on with these extremely important cells. So let's look at my first P then in this context, pheromones. So I'm going to talk about pheromones that stimulate reproduction in males in this case, right? So here is a couple of graphs of my hormone profiles. On the y-axis, you'll see blood LH concentration. So this is my brain signal, remember. And I've got 2 different rams here, 2 different males. And what I've done is I've taken blood samples up for 6 hours until the time 0 there. And then at time 0, I've introduced a very beautiful female. And the male system, the male GnRH system, responds like this. So you can see that really quickly then, the female has changed the brain signal for controlling reproduction. What about on the female side? Well, here's my female brain signal here, again with 2 different females, but here I've done a slightly different experiment. What we have here is we have female sheep that are with males for 3 months. So these are males that we call familiar males because the females are very familiar with it. They know these males very well. And when we look at the LH patterns, this is what we see. Not a lot, a couple of pulses, but you can see that it's a fairly stable situation. Now for the animal in the top here, what we've done is we've taken the familiar male, the male that she's been with for 3 months, out of the room, waited 15 minutes, and then brought the male back in again. And so when we take the familiar male out of the room and reintroduce the familiar male, nothing happens. Basically, she simply ignores the fact that this guy has gone out for a few minutes. On the other hand, if we introduce a novel male here, one she hasn't seen in this 3-month period, this is what we see. So she responds really quickly to the presence of this novel male. Now we know that this response is all to do with smell. They can smell the male. What this also tells us, though, this comparison of the familiar and the novel males, what this tells us is that females recognize and remember individual males. So we therefore need to consider memory. The memory centers of the brain are in an area called the hippocampus, in particular a band of tissue called the dentate gyrus. This is a micrograph from Penny Hawken, my colleague. Now what she did, Dr. Hawkins, she injected some female sheep with BRDU, which is a special nucleotide, and it is easily visible under UV light as a red. And she injected this BRDU just before we introduced the male. And then she killed the animals 2 hours later. And then what she saw in the memory center here is pairs of cells, 2 cells like this, where there used to be 1. So the cells have divided, made new DNA, and incorporated the BRDU into the new DNA. And then we can see the pairs of cells. So what is this telling us? It tells us that male odor stimulates cell division in the memory centers. We think, therefore, that the new cells become a new memory, and what we have now is what we call olfactory memory, the memory of smell. Okay, P2, photons. So photons is light, and light comes into the animal's brain via the eye, and it sends signals via the optic nerve to the hypothalamus and to other areas of the brain. There's some signals there, neural signals go to the pineal gland, and some other signals go to a 24-hour clock. The pineal gland produces melatonin, which is the hormone of the night, so it's only produced at nighttime. And so the length of the night is given away, is indicated to the brain of the sheep by the duration of melatonin secretion. So the longer nights produce longer periods of melatonin and tells the sheep how long is the night. The sheep brain then compares its 24-hour clock with the length of the night, and it can decide if the nights are getting longer or shorter. If the nights are getting longer, it knows it's the autumn. If it's getting shorter, it knows it's the spring. And so it tells the animal the time of the year. Then this knowledge, this information here, is used to inform the reproductive centers and tell the reproductive system what to do. In the autumn, it sends out a high frequency of generates pulses to stimulate reproduction. In the spring, it sends out a low frequency to allow reproduction to subside or to stop. So colleagues of mine, David Hazelrig and Gerald Lincoln, did another experiment which is really exciting in this area. I was not involved. They looked in the brain, at this part of the brain here, at the base of the hypothalamus, above the pituitary gland, and you can see the cross-section of the tissue here. The dark area in the center is the third ventricle. The tissue on the bottom there is the median eminence, where we have the blood vessels that drain into the pituitary gland. And you can see here, if you look carefully, I hope you can see anyway, very small green spots. So they've used the same technique that we used to study cell division in the brain. So we studied cell division in relation to pheromones. They've studied cell division in relation to the length of the night. And what happens in here, you can see lots of pairs of cells. I've blown up two here. You can see again here, pairs of cells. There used to be one cell. The cell has divided and incorporated this BIDU. They used a BIDU, which was green, whereas ours was red. So what does this tell us? New DNA is new cells. And they think that and they put a very good case here, that this is niches of stem cells, that these are cells that make new pathways, new neurons to establish a reproductive process. And they think this is part of an annual clock. So we know that many tissues and many cells have a 24-hour clock. They think that this might be a 1-year clock. Not quite 1 year, but you get the message. So what do we learn from P1 and P2 here, pheromones and photons? First, revolution number one, brain cells can divide. Brain cell division helps to control reproduction. When I was a student, this was an unimaginable scenario. The implications, we have now new thinking about how the brain works, and we have to begin to involve concepts around cell division and niches of stem cells. And stem cells in the brain give us all sorts of possibilities for the brain remodeling itself to make a new functions to control things in a different way. Also, the fact that we have stem cells and cell division means that maybe we have an avenue here for human medicine, say, where we can imagine repairing the brain when it becomes damaged. Okay, so P3 is food. Now, I'm sure your English is good enough. You understand that I'm actually playing with words here that don't really mean food with a P, but food with an F. But, you know, for the title of my talk, I did not want to have 2 Ps and 1 F. So P3, food, in males. So here we have the brain signal. In this case, it's a 96-hour period. We have to have long periods in the male sheep because they produce less frequent pulses than the female sheep. And this is what we normally see. In this particular case, 4 LH pulses, and therefore 4 GnRH pulses in 96 hours. And the experiment here is slightly different. We've gone for 24 hours here, looking at the pulses. You can see we've got 2. And then at 24 hours, we added a food supplement. So we fed the animal extra food. And this is what we saw. Basically, within about 6 hours, the brain of the sheep has begun to understand that the food supply is better than it was before, and therefore we can stimulate the reproductive system. So this is the situation we're talking about here. Food is going to stimulate this process and therefore stimulate the testes, producing more sperm and more male sex steroids. But what is happening in here? Let's look at that. So basically what happens in these sorts of experiments is we have a change in testis weight. So this is an experiment with mature male sheep that we've been feeding different diets for 9 weeks, a high diet and a low diet. And you can see what happens here is that the testis increases in size with a good food supply and shrinks with a small food supply. So we have a change in the mass of the testis. Now, every gram of testis tissue produces a certain number of sperm per day. So therefore, you'd expect in this situation the number of sperm produced per day to also be affected. And this is what happens. If we look at sperm produced per day, we see there's a large number with a higher diet and a smaller number with a low diet. Now, a bit of analysis of the data here shows us that this is an 86% increase in testis mass, but a 250% increase in sperm produced per day. So we have more grams of tissue, but also each gram has become more efficient. And so we call this spermatogenic efficiency. The spermatogenic efficiency of the tissue has increased. So how does this work? I need now to go to the situation with DNA. So as you, I'm sure, all know, right, DNA is translated into producing a messenger RNA, and then this produces a protein such as an enzyme. And this was a classic picture of DNA, RNA, and protein that I learned as a student. However, we now know that small amounts of DNA can be transcribed into what we call small RNAs, non-coding RNAs. And this RNA binds to the long RNAs and prevents the next step down the process. So it prevents the expression of the protein in response to this gene. So this gives us a new way for controlling how much protein is produced from each gene. Okay, the story now gets complicated, so I'm warning you that it's going to be a big slide and quite difficult. But there's no need to worry. I don't expect you to remember everything, just to understand the principles. What I'm trying to do is show you within the testis how nutrition and spermatozoa are connected. So here we go. Right, so please don't worry about a diagram too much. If you look across the top, you'll see central nervous system, pituitary gland, and Leydig cells. So these are the things we've sort of discussed up until now. Below that, you see the Sertoli cells, and below that, you'll see the germ cells. And what we've been doing is looking inside the Sertoli cells and inside the germ cell to see what has been happening with respect to these small RNAs. Every time you see a small yellow arrow there, this is a small RNA that has been changed by nutrition. And these small RNAs change the amount of apoptosis happening in germ cells. And this determines the quality and the quantity of the sperm that are produced. This is very exciting work, and it was done, in fact, by a Chinese student of mine, Yongjuan Guan. She came from Northwest Agriculture and Forestry University in Yangling, and she did her PhD with me, and she's now at the University of Pennsylvania. We collaborated with Lei Luo Guan in Alberta. So we have two people from China involved in this work. So it's a really interesting connection. So what did we learn from food? My second revolution. Some DNA is genes, but lots of DNA is used to control gene expression. Originally, we did not understand this, and we thought that the non-gene DNA was junk. Now we know it's vitally important in controlling the way genes are expressed. The implications, new thinking about how DNA controls cells. Clearly, this is a whole frontier area now and a very hot topic. New options as well for fixing malfunctioning tissue. So where things have gone wrong, it seems possible that we can imagine therapies for fixing the tissue using small RNAs. Okay, so heading into my next revolution, I'd like to discuss the concept we can use here with 3Ps being involved in sheep management. How can I use this information? I need to go back a step here. Basically then, I want to look at sheep products. These are the sheep products you'll see here, meat, cheese, wool. And what's happened with sheep products over the last 30 or so years is that the attitudes of societies are changing. And what this has led to is a new way of thinking. Let me give you some examples of the attitude. So this is a website called Boycott Australian Wool, savethesheep.com. And I don't want you to read the text here, just this bit. Sheep are mutilated and castrated without painkillers, then disposed of by being slaughtered in the Middle East after enduring a grueling weeks or months-long journey on overcrowded, disease-ridden ships with little access to food or water through all weather extremes. Now, I've highlighted the yellow words here because these are the words that are designed to elicit, to make an emotion in you, to make you very worried about what you're reading. Now, we can show scientifically that 90% of this is simply not true. On the other hand, 10% is. And that 10% makes us uncomfortable. In particular, sheep are mutilated and castrated without painkillers. Another website, Cow Burps Make for Bad Air Down Under. What this is saying is that cows and sheep produce 90% of Australia's methane emissions in the agricultural sector. It's true. This is a different site called Food Democracy. It's about the safety of human food. Rose has a 3-year-old son who now drinks only organic milk and eats only organic food. The amount of chemicals and hormones in our food is horrifying, says Rose, and most of the research ignores children. Now, we as scientists can work very hard to prove that Rose has nothing to worry about, that the food is safe. There will be mistakes, but we can prove that the food is safe. But she is like any mother, she's worried about poisoning her children. Importantly, Rose is the one spending the money, and she decides what she will spend her money on, what sort of food she will buy. So these 3 websites tell us that there's a change in society, that we have new market forces. These new market forces we call clean and green and ethical. Clean means that that society wants us to use less hormones and drugs. Green means that society wants us to consider the ecosystem, and ethical means that society wants us to consider animal ethics and welfare. So these market forces are now becoming part of product quality. The traditional measure of product quality was taste and odour and safety from a microbiology perspective for health. Now, the people who are buying the food want to know where it came from. What is the origin? How were the animals treated? Where were they living? What were they fed? And so on. And if we don't adopt this attitude, if we don't adopt this clean, green, and ethical concept, we risk our markets. We see that this is the future of animal industries worldwide. So let's come back to our basic question then. How do we manipulate reproduction in livestock? The current practice is exogenous hormone treatments. There's been 100 years of research and development leading us to a great range of products that I'm sure many of you have been using. These are wonderfully efficient and very powerful products, and have transformed a lot of aspects of our industry, particularly in reproductive technology. However, for most farmers, these are too expensive or not relevant to their production system. If they're involved in intensive production, or if they're involved in producing seed stock, maybe they will use them. The second thing is that they are not clean and green. They will not be seen by the consumers as desirable parts of food production. What's the alternative then? Well, the alternative is to apply the 3Ps. How do we do that? So this is the reproductive system of sheep. Very similar to goats and not too far away from cattle, but let's stay with sheep. You have a period before mating, then you have pregnancy, birth, lactation, weaning, and following that growth and development through puberty. During lactation, of course, you can harvest the milk, and when you get the animals to mature age, you can harvest the meat. So this is the production process. Now, what we want to ask is, in this situation, where is food important? Let me show you. Basically, food is important everywhere. So that's not very helpful, because we find it really difficult to supply the perfect diet to particularly grazing animals throughout the year at every stage of the cycle. So we need to do research in this area to find out when is the most important. In addition, there's another issue here, which is that food is not simply feed. Food can be looked at as energy, and in this case, energy is responsible for most of what you see in this diagram here. But in addition, food has functional nutrition factors in the diet that affect health and methane emissions. I haven't got time to go into those things today. Second thing is that we can now look at another issue here, which is time. So if you look at these two examples I've given you here, on the left is ovulation rate, and on the right is colostrum production. Ovulation rate, I put there a long green band because this is what we normally have to do for animals under field conditions. However, it is feasible to narrow this period of feed supplementation down to only a few days. So 3 days of supplementation here is enough to convince a female sheep to have 2 lambs instead of 1, 2 eggs instead of 1. Down here at the other end, just before birth, the last 7 days of pregnancy can double colostrum production. And this is really important for lamb survival. So you see here 2 very short periods of supplementary feeding that can help us improve productivity. But it's not just any 3 days or any 7 days. These are precise parts of the reproductive process. The 3 days must be a specific 3 days within the cycle. The 7 days must be the final 7 days before birth. This means you must know when these things are going to happen before you provide the supplements. Basically then, for precision nutrition, we need accurate timing. So this brings me back to P2, photons. I need to explain seasonal breeding for those of you who don't understand how the process works. As I said before, the sheep measure the length of the night, and during the year, the length of the night changes from long nights to short nights. These night lengths here, from 10 to 14 hours, are what we see in Western Australia. In response to this, the female sheep have seasonal cycles. So they have a period of time when they're ovulating and a long period of time when they're not ovulating. And we call these the breeding seasons. Breeding season, non-breeding season, and a second breeding season that follows. Now, what is interesting here is that we can introduce pheromones. Now, I showed you how the pheromones of the male sheep can switch on the reproduction in the female sheep. So when we introduce rams here, novel rams, remember, we have every female ovulates at the same time. They all smell the male at the same time, all of the females in the flock, and they all respond at the same time. So we have synchronized ovulation in the flock, therefore synchronized pregnancy and synchronized birth. And this synchronization gives us real power over the management of nutrition during the reproductive process. So down here then, pheromones can help us to control when is day 0 and therefore the processes that follow. Now, let's go back to the male side here, because what happens is, of course, it's not just the females that have a seasonal cycle. The males also have a seasonal cycle where the testis grows and gets smaller, then grows again. And of course, this is associated with an increase in testosterone production. And because testosterone controls the pheromone, it controls the amount of pheromone produced. So what happens here is then that testosterone in the male controls pheromone production to induce ovulation in the female. And P2 photons controls the amount of testosterone being produced. So basically then, if we add a light signal, we can increase the amount of testosterone produced. And we can do that to get a stronger stimulus to actually improve the efficiency of the system by manipulating the length of the night for the male animals. This is a relatively simple thing to do because in most flocks there are only a few male animals. You don't have to have a very big room with light control to make it happen. And I have a colleague in Mexico, Jose Delgadillo, who does this with goats for his goat farmers in northern Mexico. He puts the males into a building and controls the length of the night to help improve this response to pheromones. So basically then, photons give us the control of the length of the night and allow us to improve the pheromone response. So using the 3Ps is what we can do here to control this process, to improve the efficiency of it and to control the timing. And we don't need to use any drugs or chemicals, as you can see. So this was my third revolution. We now know we can control reproduction in animals by manipulating their environment. The implications for this: clean, green, and ethical management of livestock. And then, of course, I think application worldwide. So let me summarize where I've been. Pheromones, photons, and food are my 3Ps. My starting question when I was a young scientist was, how does the reproductive system respond to changes in the environment? Revolution number 1 was brain cells can divide. As I said before, when I was a student, this was just not thought possible. Revolution number 2, some DNA is genes, in fact, not very much as genes, but lots of other DNA controls the gene expression. And revolution number 3 is we have a brand new vision for livestock management. Thank you very much for listening.